Hello guys, today in this video we're going to start the part of the IMAT which is called Bioenergetics, and we are going to cover the energy currency of cells, ATP, and glycolysis, aerobic respiration, and fermentation. In the next video we're going to cover the redox reaction and photosynthesis. Alright, so let's get going right away. First of all, energy. Why do living organisms want energy? All living organisms require a continuous supply of energy to stay alive. Where does that come from? Either from the absorption of light energy or from chemical potential energy, energy stored in nutrient molecules. The process of photosynthesis transfers light energy to chemical potential energy, and so almost all life on Earth depends on photosynthesis, either directly or indirectly. Photosynthesis supplies living organisms with two essential requirements: an energy supply and usable carbon compounds. All biological molecules, such as carbohydrates, lipids, proteins, and nucleic acids, contain carbon. All living organisms therefore need a source of carbon. Where does it come from? There are, according to where the organism takes carbon, we have two categories: autotrophs and heterotrophs. Organisms that can use an inorganic carbon source in the form of carbon dioxide are called autotrophs. Those needing a ready-made organic supply of carbon are heterotrophs. An organic molecule is a compound including carbon and hydrogen. Originally, this term meant a molecule derived from an organism, but now it includes all compounds of carbon and hydrogen, even if they do not occur naturally. Organic molecules, can be used by living organisms in two ways. They can serve as building bricks for making other organic molecules that are essential to the organism, and they can represent chemical potential energy that can be released by breaking down the molecules in respiration. This energy can then be used for all forms of work. Heterotrophs depend on autotrophs for both materials and energy. Alright? Now, we all have said that the energy currency in all cells is ATP. But what is ATP? Let's look at the structure first of all. A molecule of ATP, or as it is called, ATP adenosine triphosphate, consists of adenosine and triphosphate. What is adenosine? Adenosine is the nucleotide adenine plus ribose. So this is adenosine. And triphosphate stands for three phosphate groups. 1, 2, 3. So adenosine, adenine plus ribose, and triphosphate, three phosphate groups. You see, ATP is called an unstable molecule because the three phosphates in ATP are all negatively charged and repel one another. So when a phosphate group is removed from ATP by what procedure? Hydrolysis, a more stable molecule, ADP, adenosine diphosphate, because it has only two phosphate groups, diphosphate, ADP, is left, which is a more stable molecule. So the change from a less stable molecule to a more stable molecule always releases energy. And this is how ATP provides energy for all cells. So it breaks down, it gives one phosphate group, and by giving one phosphate group, it releases energy, which is used for another reaction to happen. So these bonds here between the phosphate groups are called high energy bonds because they have energy, and when we break the bonds, this energy is released from the for the cell to use them in their reaction. And this is why the ATP is the energy currency in a cell. To be more specific, when a phosphate group is removed from ATP and ADP is formed, 3.5 kilojoules per mole of energy is released. Removal of a second phosphate produces adenosine monophosphate, AMP, and 30.5 kilojoules per mole of energy is again released. Removal of the last phosphate, leaving adenosine, releases only 14.2 kilojoules per mole. In the past, the bonds attaching the two outer phosphate groups have been called high energy, because more energy is released when they are broken than when the last phosphate is removed. But this is in order to understand it. It's not actually what happens. So this term, high energy, can be a little misleading. So I urge you to avoid that term. Why? Because the energy does not simply come from breaking these bonds, but rather from changes in chemical potential energy of all parts of the system. Now, of course, all these reactions are reversible. That means that ATP can get to ADP plus phosphate, and again ADP can connect with the phosphate and create ATP. Okay. Okay, now let's see about cell respiration. Cell respiration is the means by which cells extract energy stored in food and transfer that energy to molecules of ATP. So what happens? What is the final goal of respiration? To create ATP molecules. What does that mean? Let's take one adenine and one ribose and phosphate groups and connect them all together. This is what happens during respiration? No. So we said that the cell needs ATP in order to break the bond here, to make this reaction happen, ATP to give ADP plus phosphate, to supply energy for metabolism. But what happens with these two? They are left together, free. So in order to have energy for more reactions, these two need to reconnect and give ATP. This is what respiration does. It takes the energy from the food to give it to synthesize ATP. And then by desynthesizing or breaking down ATP, we have the energy to use to metabolism. This is what these cats represent. Okay. So energy that is temporarily stored in molecules of ATP is instantly available for every cellular activity, such as passing an electrical impulse, contracting a muscle, moving cilia, or manufacturing a protein. The equation for the complete aerobic respiration of one molecule of glucose is a highly exergonic process. That means that it releases energy. And now there are two types of cell respiration, anaerobic and aerobic. The word aerobic comes from the Greek word aero, which means air. Aeras in Greek means air. And what is the main constituent of air? Oxygen. So when we say aerobic, we mean oxygen present. And when we say anaerobic, we mean oxygen absent. Okay? An is this two-letter syllable that shows the negative part of this word. Okay, so aerobic with oxygen, anaerobic, the negative of this word, no oxygen. Okay, so if oxygen is not present, anaerobic respiration, what are the steps to have ATP? We've got glycolysis, the first step, and then alcoholic fermentation or lactic acid fermentation. We see what this is. If we've got oxygen, the first step is glycolysis, then we've got the link reaction, and then we got the Krebs cycle, then the Electron Transport Chain and oxidative phosphorylation, which happens almost together. Okay, and we will see each of that separately. So first of all, we'll need to talk glycolysis because it's the first step in both. Okay, what is glycolysis? Glycolysis, I believe I'm calling it correct. Lysis, here, glycolysis says lysis. The Greek word leo or lino means I break something down, I split. And glyco comes from glucose. So glycolysis, what is the splitting or lysis of glucose? It is a multi-step process. And actually it is a 10-step process that breaks down one molecule of glucose, which is how many carbons does glucose have. Let's remember our biochemistry lessons. Just remember the formula for glucose. It's C6H12 oxygen 6. So it takes glucose and it breaks it down. Glucose is a molecule with 6 carbon atoms, and it is split into 2 molecules of pyruvate. The resulting the result product from the 10-step process is pyruvate, which is a 3-carbon molecule. Okay. Energy from ATP is needed in the first steps, but energy is released in later steps, which can be used to make ATP. There is a net gain of 2 ATP molecules per molecule of glucose broken down. That is why. The activation energy for glycolysis is 2 ATP. And in the last steps of the 10-step process, we've got energy to create 4 ATPs. So we create 4 ATPs. We used 2 ATPs, therefore the net gain for the cell is 2 ATP molecules. Okay. Glycolysis takes place in the cytoplasm of a cell. And let's see a diagram here. Of course, you don't need to know the steps or the enzymes or the compound names. I want you to see that the end product is a pyruvate. Okay. I want you to know that glycolysis produces ATP without using oxygen, and that each step is catalyzed by a different enzyme. And although this process releases only 1/4 of the energy stored in glucose, it is critical because the end product, pyruvate, is the raw material for the Krebs cycle. Actually, pyruvate is used in the link reaction to give me something else, which we'll see is very, very important for Krebs cycle to take place. And okay, I will stop talking now. Let's see here some things that I want to show you. First of all, during glycolysis, ATP is produced by substrate level phosphorylation. That means by direct enzymatic transfer of a phosphate to ADP. Okay, so why is that? Because glucose is an energy-rich molecule, but does not react easily. To tap the bond energy of glucose, energy must first be used to make the reaction easier. 2 ATP molecules are used for each molecule of glucose to make the reaction easier. Two ATP molecules are used for each molecule of glucose to make first glucose phosphate, then fructose phosphate, then fructose biphosphate, which breaks down to produce 2 molecules of triose phosphate, you see here. but I don't want you to know that. Okay. Now we're watching these here, and we are going to watch them a lot. What are these molecules? NAD and NAD hydrogen, or NADH, or reduced NAD. What are these? Have we ever talked about them again? No, we haven't. Usually, most books, they keep talking and using them. They say glycolysis, anaerobic respiration, aerobic respiration, and after they finish all the theory, electron transport chain, everything, then they tell you what NAD and reduced NAD is. And this is how I have constituted my presentation file here, the PowerPoint file. But I won't constitute the video the same way, because in order to understand the next steps, I want you to have a rough idea what these are. So let's get into what these are here. NAD and FAD. They are required, okay, for normal respiration. They are coenzymes. This is what I want you to know, that they are coenzymes that carry protons or electrons. That's their function. So we've got NAD, nicotinamide adenine dinucleotide, which is made of two linked nucleotides. And if NAD takes, this is what we're going to see, the oxidized form of this, takes two hydrogen, it becomes this. And this reduced form, it carries one proton and two electrons. Let me make you understand this. If you remember the hydrogen structure, it's like that. This is the nucleus. It has one proton here. Let me write that with P plus, one proton and one electron around it. Here, electron. All right. This is hydrogen. Hydrogen plus is what? The ion or cation of the hydrogen that has lost one electron. So what is left? With a proton. Hydrogen plus is one proton. Okay, so the NAD H, the reduced form, it carries one proton and one electron because it has a hydrogen, and the plus sign, which is one. I'm sorry. No, I'm sorry. Let me start again. The NAD, this, the oxidized form, receives two H's, except one proton. Two H's, these, have two protons and two electrons. Minus one electron, one proton, sorry, this is one proton. What are we left with? One proton and two electrons, okay? So it is an proton and electron carrier. NAD P is the same, it's a hydrogen carrier molecule. And FAD, again, it has an oxidized form and a reduced form. So the oxidized form does not carry hydrogen or electrons or protons, but it is ready to accept them, and the reduced form carries them. So what happens during here glycolysis is that the oxidized form of NAD is taking electrons, and we have the result of NAD, the reduced form of NAD, NADH. Okay. Now I need you to go back to the enzyme video in biochemistry, where I talked about allosteric enzymes and allosteric inhibition, what it is. I have explained it in the past, but now we'll show you how it is being used here. The end product the third enzyme PFK, this phosphofructokinase, PFK, is an allosteric enzyme. That means what? That it can be inhibited by something. That means that a substance can get to it and not allow it to do its job. What is this substance? ATP. So PFK can be inhibited by ATP. Is that important? Yes. Because this is how glycolysis is being stopped. So when the cell has a lot of energy, a lot of ATP molecules, the ATP will bind to the PFK and won't allow it to do its job. Therefore, the glycolysis will stop and no more energy will be produced. Is that understandable? Again, go and look the video. If you don't remember what I'm talking about, what is allosteric inhibition, go and look at the biochemistry video. I believe it's the second or the third one, where I talked about enzymes and proteins. I have talked about that and I have an example there. Okay, let's move on now. Okay, I've talked about that. We have modes of regulation of glycolysis. Not only that, not only ATP, but we've got positive and negative allosteric inhibition. That means positive, if we've got pyruvate kinase, this product allows it to act more, and negative, it allows it to stop acting. So there's much type that it is about biochemistry level, university level biochemistry, that you're going to need to know that. So I'm not going to move on and discuss about that. Now, we said that in anaerobic respiration, the first step is glycolysis, and what follows then is fermentation. Anaerobic respiration and fermentation is not a synonym for glycolysis. Anaerobic respiration consists of glycolysis plus fermentation. What is it? It is an anaerobic catabolic process. Okay. That consists of glycolysis plus alcohol or lactic acid fermentation. It originated as a process millions of years ago when there was no free oxygen in the Earth's atmosphere. Even today, it is the sole means by which anaerobic bacteria, such as this one, let me make an effort to call it botulinum. I have no idea if I'm calling it correctly, if I'm pronouncing correctly, so feel free to correct me anytime. It's a bacteria that causes a form of food poisoning, botulism, and its means of producing energy is mainly anaerobic. Only anaerobic, actually. There are facultative anaerobes that can tolerate the presence of oxygen, and there are obligate anaerobes which oxygen for them is toxic and they cannot live, like Clostridium is a very common family of bacteria. Now, fermentation can generate ATP during anaerobic respiration only as long as there is an adequate supply of NAD, the oxidized form of NAD, to accept electrons during glycolysis. Without some mechanism to convert reduced NAD back to oxidized NAD, glycolysis would shut down. Fermentation consists of glycolysis plus the reactions that regenerate NAD plus the oxidized form of NAD in order to be ready to take the electrons back, to accept electrons. And we've got two types of fermentation, alcohol and lactic acid fermentation. Let's start with alcohol fermentation. Irreversible fermentation is the process by which certain cells convert pyruvate, here, let me show you that, convert pyruvate, the end product of glycolysis, into ethyl alcohol, acetaldehyde, I'm sorry, I said the Greek term, acetaldehyde, and carbon dioxide, in the absence of oxygen. And in the process, they oxidize the reduced form of NAD back to the oxidized form of NAD. The break-making industry depends on the ability of yeast to carry out fermentation and produce carbon dioxide. The carbon dioxide causes bread to rise. The beer, liquor, and wine industry depends on yeast to ferment sugar into ethyl alcohol. Now, lactic acid fermentation. No, I've got no picture for that. During that, pyruvate from glycolysis is reduced to form lactic acid or lactate. This is the process that the dairy industry uses to produce yogurt and cheese. Also in the process, NAD8, that is the reduced form of NAD, gets oxidized back to NAD+. Human skeletal muscles also carry out lactic acid fermentation when the blood cannot supply adequate oxygen to muscles during strenuous exercise. Lactic acid in the muscle causes fatigue and burning, and this is why after some time of strong exercise you may get cramps. The reason why you get cramps is because of lactic acid. Okay, the lactic acid continues to build up until the blood can supply the muscle with adequate oxygen to repay the oxygen debt. With normal oxygen levels, the muscle cells will revert to the more efficient aerobic respiration, and the lactic acid is then converted back to pyruvate in the liver. Moreover, if you eat a banana, because it has potassium, potassium reacts with lactic acid, and it does not allow you to have cramps. And this is the process of anaerobic respiration. Now let's see, yes, I have forgot to put a title here. Now we start what happens with aerobic respiration. And anaerobic respiration, the steps are as I'm telling you. First, we've got glycolysis, which we get the pyruvate. Then, through the link reaction, the pyruvate begins this molecule. And then, this molecule is needed for the Krebs cycle to take place. And then we've got other processes as well, but let's start here. So we've got glycolysis. We get what? Pyruvate. Okay? Pyruvate now passes by active transform from the cytoplasm through the outer and inner membranes of the mitochondria and into the mitochondrial matrix. Here it is decarboxylated. What is that? D means I get something far away. Carboxylated, it has the word carbon. That means that carbon dioxide is removed. And dehydrogenated, that means that I take away D and hydrogen, so hydrogen is removed, and combine with coenzyme A to give me a substance called acetyl coenzyme A or acetyl CoA, as I will be calling it from now on. The hydrogen removed from pyruvate is transferred to the oxidized form of NAD. Okay, this is known as the link reaction. Coenzyme A is a complex molecule composed of a nucleoside, that means adenine plus ribose, with a vitamin here, which is called pantothenic acid, and acts as a carrier of acetyl groups to the Krebs cycle. The hydrogen removed from pyruvate is transferred to NAD. Okay, do I have the reaction somewhere? No. Now, fatty acids from fat metabolism may also be used to produce acetyl CoA. Fatty acids are broken down in the mitochondria in cycle of reaction, in which each turn of the cycle shortens the fatty acid chain by a 2-carbon acetyl unit. Each of these can react with CoA, coenzyme A, to produce acetyl CoA. which is like that produced from pyruvate, now enters the Krebs cycle. So let's get into here, the Krebs cycle. What is it? It was discovered back in 1937 by Hans Krebs. It's a closed pathway of enzyme-controlled reaction. What happens here? Acetyl-CoA here. It combines with a 4-carbon compound, 1, 2, 3, 4, to form a 6-carbon structure. 1, 2, 1, 2, 3, 4, 5, 6. Citrate. Okay. So I want you to know that the first reaction of the cycle is oxaloacetate, which combines with acetyl-CoA to give me citrate. Only what a circle is what I believe you should know from here. Then the citrate is decarboxylated and dehydrogenated in a series of steps to yield carbon dioxide. So I got a waste product. What is my waste product? Carbon dioxide. 2 molecules of carbon dioxide as my waste product. And hydrogens which are accepted by the carriers NAD and FAD. So I got 2 NAD, 2 reduced NAD molecules. I take one more, 2 reduced NADH and 2 reduced FAD molecules. I'm sorry, no, 1, 2, 3 reduced NAD molecules and 1 reduced FAD. So, for each turn of the cycle, 2 carbon dioxide molecules are produced, 1 FAD and 3 NAD molecules. This is a mistake. Yes, I apologize. I need to correct it. This is 1. Let me write it down for a second to correct it. I'm sorry, I apologize for that. 1 FAD, H, 1 reduced FAD is the end product. So, what's that? Let me give you a little bit more knowledge about the Krebs cycle. First of all, I want you to know that it happens in the matrix of mitochondria. Okay. It turns twice for its glucose molecules that enters glycolysis, 1 for each pyruvate molecule that enters the mitochondrion. The cycle generates 1 ATP per turn by substrate level phosphorylation, but most of the chemical energy is transferred to NAD and FAD. The reduced coenzymes, NADH and FADH2, shuttle high-energy electrons into the electron transport chain in the crystal membrane, which we will see right now. The electron transport chain. In the final stage of aerobic respiration, oxidative phosphorylation, the energy for the phosphorylation of ADP to ATP comes from the activity of the electron transport chain. This takes place in the inner mitochondrial membrane, which is called the crystal. Now I have the steps here, I have the theory here, all of it, and everything that happens, but this is for your own study. Okay, these are for your own study. We will look at this and see what happens. Here, it's the mitochondria. This is the matrix, and this is the intermembrane space, because you remember, let me remind you the structure of the mitochondrion. It's like that. It has one outer membrane, and it has one membrane internal that makes these types of folding in order to increase its area. This is called the crista, if you remember, and this is the intermembrane space. And this is the matrix here. Alright? Let's start. First of all, reduced NAD and reduced fat are passed to the electron transport chain. What happens? The hydrogens are removed from the two hydrogen carriers. We said that the NADH has hydrogens. So the first step is to take these hydrogens from the hydrogen carriers, and it's split into its constituent proton and electron. So we take the hydrogen and we split the hydrogen to protons and electrons. The energetic electron is transferred to the first of a series of electron carriers. Most carriers are associated with membrane proteins, of which there are 4 types. A functional unit called a respiratory complex consists of one of each of these proteins, arranged in such a way that electrons can be passed from one to another down an energy gradient. That means electron here, second, the third, and the fourth, it passes through an energy gradient. That means from what? From a higher energy level to another one at a lower energy. And during this passing, we've got what? The release of energy. Okay. Some of this energy is used to move protons from the matrix of the mitochondria. Here we've got protons, you see? From the matrix into the intermembrane space, the space between the inner and the outer members of the mitochondrial envelope. These produce a higher concentration of protons in the intermembrane space than in the matrix, setting up a concentration gradient. So we've got a high proton concentration here. Therefore, by diffusion, they want to pass where? Inside the matrix, and they do. Now, protons pass back into the mitochondrial matrix through protein channels in the inner membrane, moving down their concentration gradient. So from the intermembrane space, so we used energy to get them to the intermembrane space, and now they pass themselves back into the matrix. Now, this channel is associated with an enzyme, this blue big one, which is called ATP synthase. Let me show you that word. Yes. Does this word remind you any word? Yes, of course, it reminds you the word synthesis, which means make off. So this is an enzyme that makes ATP. What does this enzyme use? It uses, as the protons pass through the channel, their electrical potential energy is used to synthesize ATP in the process called chemiosmosis. Here. Okay. Finally, oxygen has a role to play as the final electron acceptor. In the mitochondrial matrix, an electron and a proton are transferred to oxygen, reducing it to water. And now the process of aerobic respiration, an electron and a proton, an electron and a proton, that means water, one hydrogen plus one oxygen gives us a water molecule. The process of aerobic respiration is complete. The sequence of events in respiration is wisely used. Why? Because we've got ATP production, and now we've got a serious number of ATP molecules made. (28) ATP made during oxidative phosphorylation. Let me show you some more numbers. Theoretically, 3 molecules of ATP can be produced from each molecule of reduced NAD, and 2 molecules of ATP from each molecule of reduced FAD. However, this yield cannot be achieved unless ATP and phosphate groups are available inside the mitochondria. About 85% of the total energy yield of electron transfer is used to transport ATP into the mitochondrion and ATP into the cytoplasm. Hence, each reduced NAD molecule entering the chain produces on average 2.5 molecules of ATP, and each reduced FAD produces 1.5 molecules of ATP. The number of ATP molecules actually produced varies in different tissues and different circumstances, largely dependent on how much energy is used to move substances into and out of the mitochondria. Alright, now I want to have, oh dear God, what's that? No, no, where is my picture? Oh yes, that's correct. I was scared for a minute. Alright, let me have a summary. Okay, first, what's happening? This is respiration, how it's happening. We've got glucose that comes into the cell, in the cytoplasm, and this is where glycolysis takes place. And we've got the end product pyruvate. Then pyruvate is being transferred into the matrix of the mitochondria, and through the link reaction it gives us some CO2, and the most important, it is being combined, the pyruvate here, with coenzyme A to give us acetyl-CoA. This acetyl-CoA is being combined with a 4-carbon substance, oxaloacetate, to give us, I'm sorry, I forgot, citrate, a 6-carbon substance. This, through a series of steps, is doing what? It is giving protons and hydrogen to reduce NAD and FAD. Actually, we've got the end production of reduced NAD and FAD. This is being used in the electron transport chain. What exactly happens here? The NAD and FAD are being split into what? Their protons and electrons. Their protons are being passed through energy into here, and then, because of the concentration gradient, they are passed through the ATP synthase again back into the matrix. But during passing the matrix, they supply the energy needed for the synthesis of ATP. Also, the electrons that was remained by the splitting of these, and the protons that pass through here are being combined with oxygen molecules to give us water. And this is the aerobic respiration, and it is a very important process, because it is doing what? It's a very important process because it in okay, I forgot what I wanted to say. Oh yes, because it creates ATP, and we said what we mean creates ATP. It connects again phosphate groups to the ADP in order to have the energy inside the ATP, for the energy to break it and take the energy wherever it needs. Okay, now what I forgot to say is about cytochromes, which I have it here somewhere. Yes, the ETC, the Electron Transport Chain, it consists of cytochromes. These are protein molecules, similar to what? To hemoglobin. We have seen the structure of hemoglobin, and cytochromes are present in all aerobes, but they have differences according to the evolution state of the organism, and we are using them to oh my god, we are using them, the cytochromes, to trace evolutionary relationships. Okay, is there anything else I would like to say before I end this video? Yeah, let me show you one last thing. No, actually, that's all. Also, I wanted to say now that I will create a test and I will upload it in all the videos of the cell combined. There will be a test in the last the cell video, and I will make a video about the sanction bioenergetics and I will finish it, but I won't make a videos about the section reproduction and inheritance. No, this is a waste of time for me right now because I'm studying as well, and I won't make videos about that topic. There are many videos you can watch about that, and after September 14th, that I will have some time, I will make videos about that. So only for guys who are giving this year won't have these videos, but they will be uploaded sometime. I'm really looking forward to start to upload the videos about anatomy and physiology, because I believe no one has something specific to study, and you either study too much or too little about that. And I'm really interested to start making videos about that, anatomy and physiology of animals and humans. And around August 20th, and maybe a little bit later, I will upload videos about oh god, I will upload videos about chemistry. I will start, yes, making chemistry videos. That's it. I have nothing else to add. Thank you so much. Bye bye, guys.